welcome back to the lecture series on cell culture. So, we are into the week 3 and we have finished our first class. So, we are into the second class. So, in the last class, if you recollect, we were talking about the microscopy. So, we have talked about the challenges of or the need for an upright as well as an inverted system, and there is something in the end I told you about the numerical aperture. There is something which will be written on the lens. So, if you see an objective, if you something like this, okay, or objective like this, or you can, and you will see the this is where you fit the objective. So, you have all the grooves which are cut there, or if you see it from the top and something like this, okay, or you are seeing it like this. Something like this. So, out here there are some small things which most of us kind of, you only look for it is written 40x or 10x or 20x, like with Swan and so forth. But underneath that you will see something very small written like n, which is essentially small n, which is essentially called the numerical aperture. It's called numerical aperture. This numerical aperture is the one which will tell you whether this objective is best suited for viewing through glass or through plastic or some other material. I am not getting into the physics of it at this point because this is not within the purview of it, but those of you who are interested, you can open up and check how that numerical aperture is being, because it is a property of the lens itself inside the objective, and this is something, it's my experience that every time the big giant microscope sellers kind of do not tell you about it. They just wanted to push their stuff. So, be very careful on this, because these objectives, most of the top companies, whether you talk about Zeiss, Nikon, Olympus, all of them charges a fortune for individual objective. Everything is in somewhere in the range of say 1 lakh or something as individual objective. I am not even talking about the microscope. Microscope budget goes anywhere between a standard good quality microscope, if we talk about, somewhere between say 15 to 30 lakhs. This is the kind of budget we talk about, good quality microscope. You can get some of the cheaper version, unless, I mean, they may not be the best, but you can work out with them within a range of say 5 lakhs. But whenever you have to plan for a microscope, you have to really plan in advance. Whether even 5 lakhs is not something which is like you ask for it and you get it. So, you have to be very careful about planning out how really you want this stuff to be. So, again, you don't want to mess up with it. So, if you have two kind of samples, say for example, you have a plastic sample, you know that you will be plating your cells on top of a plastic, and you have a glass vessels, then you may need to have two different kind of objectives with different numerical aperture on the same microscope. That's not an issue, but you have to have different objectives. Now, suppose there is a third thing about the microscopy. If you want to, if you are working, especially there are several labs sitting in IIT, I know there are a handful of labs who work on metallurgical sample for cell related study, or some people work on electrodes, integration of cells on top of electrodes. For them, or they may work on microelectrode arrays and field effect transistor chips. Those are completely opaque systems. You may opt for another version of upright, which is a metallurgical microscope. You may think over that, but again, so just think it's there. Metallurgical microscope. So again, you have to realize couple of stuff. It is a need, a requirement, and most importantly, as I told you, it's about the budget. What are you talking about? So budgetary constraints will always be there. So you have to ensure that within your available budget, you are playing your game. So that's why I told you in the beginning that let's have a roadmap, like I'll achieve this by this time, this by this time. Likewise, unless you have a proper roadmap, you'll stumble upon consequences which you do not want to get into. And this is, I'm talking from my soul, after at least developing 5 to 6 independent state of the art facility across the world, that I have gone through that roller coaster ride, that lab has this much fund, so okay, now we have to restrict. But in the very early phases, I had no one to tell me that it's a job, so okay, let's get this, but did not kind of take into account that what, here you are going to face a roadblock. What you are going to do? So please plan things neatly, and you should have a margin, like after 1 year I will be getting this, after 2 years I'm going to be getting this, after 3 years I'm going to. So if you have a layout plan for 10 years, you are good to go. Now with the microscope, there is another aspect which adds up. Do you want, so these sellers will tell you, do you want a provision to attach fluorescent facility, or do you want the fluorescent facility? Because most of the modern day cell biology depends heavily on different kind of antibody conjugated markers, or people use nanoparticles, fluorescent nanoparticles, different kind of conjugations, different kind of histology, where you have these markers, different kind of calcium binders, different kind of movement or the drift of different kind of ions. So I, on a slight biasness, will recommend go for a fluorescence if your budget permits. So in other words, then get a really good microscope where you have integration of the fluorescence. And again, with the fluorescence, you will come across an interesting thing, which all wavelength you will be getting. Most of them will come for, you will be able to visualize anything which is fluorescing red or green, but you may not be able to visualize something which fluoros blue, because those lasers are costly. So again, these are some of the points what you have to take into account. Those laser diodes which are there for the blue are costly as compared to red or green, or you may even go for a far red. So there are 4 zones where you can play. You have a UV, for blue, you have, if you put it like this, it will make easy. You have blue, you have UV before that, okay. You have green, you have red, and you have far red. This is the kind of zone where you are playing this game. So be careful whatever you are asking. Whatever you will ask, you will get that. So this fluorescent assembly is a costly affair. But as I told you, with my biasness, I will recommend you have a provision for adding fluorescence. You will be benefited by it. Do not leave it, because this is needed. Any lab, wherever you set up, it's a chemical biology lab or a bioengineering facility or a tissue engineering facility or a developmental biology facility or a biomaterial cell interface facility, fluorescent will be essential. So do not become frugal on it. Instead, just get it. Okay. So keep in mind what kind of laser diodes you are getting, and if you have any confusion, grab a proper manual on microscopy, read through them, and all those very in nutshell what I have told you will be very clear to you what I am trying to tell you. And much of the things what I am telling you are the practical hurdles you are going to face, which the books will tell but you will overlook most likelihood, and the seller will never tell you, because seller wants to sell his stuff. They are not going to tell you that, you know, this is the problem you are going to face with this, this is the problem you are going to face with this. They will just, you know, ensure that you buy this stuff, and then they will ask you, oh, you have to add up this, oh, you have to add up this. So don't fall in that trap. So these are some of, I may have missed out something, and if I recollect during the window of this course, I will definitely put it down there. But these are certain things what in practical day-to-day life I have faced, and I will not wish you young people face that problem. Now from here, we'll talk about few other instruments which you'll be needing pretty dearly in your facility. One of the things which you'll be needing in both sides, here also, will be a pH meter, because whenever you are dealing with any kind of medium, any kind of buffer system, you have to have a pH meter, absolute essential. Buy a good local quality pH meter and keep it in a safe corner or safe place where it doesn't fall down. Put it in a stand and ensure that it's the standards, because the pH meters comes with the standards, like, you know, you'll have pH 1, pH 7, pH 10, likewise you'll have standard bottles in it. They are kept properly closed, and you have a kind of a spot where you can measure it. Next in that line, apart from pH meter, will be another instrument which most of the labs I visit time to time do not carry, but something which is very, very important. That is called osmometer. Osmometer. So, osmolarity. In a layman language, that means, suppose here I have a bottle and this is fluid, and it has certain other kind of particles in it, and the green ones are showing the particles, and these particles generates a pressure, okay, which is part of something you guys must have studied called colligative properties of material. Osmometer is being used using something called a freezing point depression process. Again, I am not going to get into that freezing point depression process because it falls under the studying the colligative property. It will give you how much proportion, in a simplest language, how much proportion of solute is present there. And if you talk about our body fluid, it hovers around 320 milliosmoles. That's essentially your extracellular fluid. As compared to, if you look at your cerebrospinal fluid, which is at least 100 magnitude lesser than that, it is around 220 milliosmoles. Why these values are important and why am I taking the pain to tell you this once again? Okay. So, that means, suppose you are working on developing a medium for neural culture, neurons, right? So, neurons are, if you see this picture, so for example, you want to culture the hippocampal neuron, okay, or cortical neuron, or any part of the brain. So, these are protected by a blood-brain barrier, BBB, and they are bathed in cerebrospinal fluid, CSF, cerebrospinal fluid, blood brain barrier. Okay. So, if you are developing a medium for the neuronal cells, then you should know that what is the milliosmolarity. So, most of the neurons grows better between 220 to 230 or even up to 260 milliosmoles, as against the other cell types of the body which are in direct contact with the blood, which are around 320 milliosmoles. So, many a times your cells dies, especially fragile cells like neurons or neuron derivatized cells, because your cell has a very high osmolarity or you do not see the proper growth. So, for example, once we will go to the medium, you will realize medium like DMEM, okay, all these have F12, HAM, these are different medium names, they all have pretty high osmolarity values, as compared to some of the mediums in recent times what have been developed, like neurobasal, exclusively developed by Professor Brewer in Southern Illinois Medical School. These are hovering around 220 to 15 milliosmoles. So, this is something which time to time you may need to check that what is the osmolarity value of the medium what you are using. So, you have to just take, like in a Eppendorf tube, you have to take a small sample of the medium in a sterile manner and you just test it. I mean, this device, it's a very interesting colligative property and it's fun. So, this is kind of to keep track. So, you have to have a spot where you will be putting the osmometer. Okay. So, see, now slowly we are covering the space what we have dedicated for the countertop. So, now you have a pH meter, you have an osmometer. Now, we needed something what I haven't mentioned, you will be needing a somewhere, because you have to thaw the cells. If you are using cell lines, you have to thaw the cell lines. If you are using some frozen medium, you have to thaw it. So, the best idea is to have a small water bath somewhere in the, again in the corner. You have to be careful wherever you keep a water bath, because there are a lot of water spill happens at that zone. So, either, and second thing, forgot this, before I forget this, you have to have a sink close by. So, you needed to curve out another sink somewhere out here in the inner cell culture facility, a small sink, and adjacent to the sink you have to have a water bath, because you have to fill this water bath. So, you do not want to carry water in something and go and pour on it. Or you have other option is that you keep a water bath somewhere out here, outside. You may need one. So think over it, what kind of water bath you are going and where you want to place the water bath, because you'll be needing water bath. So in that case, you needed to have a sink along with a water bath adjacent to it. That way you will be ensuring that you are not spilling out things. And it is always a good idea to have another sink inside. May not be a bad idea, because you are working, you want to wash your hands, you want to go to the next room, or you are coming from the next room to inside, wash your hands. Or if you want to really minimize, you can have one sink. Again, these are all function of the amount of space you are having, right? Without that, I mean, it is tricky. So think over it, what all you needed. So definitely be needing a water bath. That is for sure. There is no question on that. So now the thing is that either you can get away with one water bath out here, somewhere out here, you can place a water bath, or you can have to have a water bath here. But in that case, you'll have to travel back and forth like this. Okay. And one more thing which I forgot is, while you are working out or kind of, one second. Okay. While you are wrestling through using or buying a camera, so the companies which you may like to look forward for good quality cameras will be Zeiss, Nikon. You may even think of Olympus, but again, it's all about your budget, where you are hitting upon and how far you want to hit on this. And one more thing, do you want a camera on top of your microscope? Again, out of my farsightedness, I will recommend go for it, because that is really, really helpful. Because then you have a recording. Like say for example, you have a sample, you want to take wonderful fluorescent picture. You'll be doing later some confocal microscopy or something, but you want to take that picture. Or your cells adhering to some fluorescent nanomaterial or some other material. So it's always a good idea to have the camera. And with the camera also, you have to be careful which company's camera you're going to go through. How you're going to fit the camera, what will be the camera resolution and what. So there are companies like Hamamatsu. They really sell good quality camera. But then the very moment you are having a camera into the system, you have to have a monitor adjacent to you, because you want to take pictures now, right? So your microscope will have a setting adjacent to it, a computer setup. And then, of course, the seller of the microscope will provide you the software, and you have to have an image processing software along with it. So you are seeing how the price is continuously jacking up. And these are, I'm telling you, these are bare essential when you are competing or when you are trying to pull out really good quality work. So that's why it becomes very much more imperative that day zero, when you start to build up the facility, you think twice. Ask some of the colleagues who knows it. Discuss among the user group who will be using it. It is very, very, very essential. Think through it thoroughly. Okay. I'll close in here. In the next class, we will add up some new paradigm into this, what will make it a much better and much more beautiful facility. So let's recollect what all we have talked today. We have talked about some of the fundamental information about the objectives. We have talked about the need for an osmometer. We have talked about the need for multiple pH meters. We have talked about water baths which has to be kept there, and in the end we talked about to have a camera setup along with the basic setup of the microscope, and the need for having a monitor along with image processing softwares, which partly will be provided by the microscope makers, and rest you may have to have a Photoshop or something installed in it where you transfer the images, or if you want to make a video, what all facilities you have. Okay? Thank you.